Welcome. Good afternoon, good evening, or good morning, depending on where you're tuning in from. I believe we have participants with us today from Europe, from Asia, and the United States. So welcome, I'm Robert Keeval, the CEO of IMMR's US headquarters in Minneapolis. And today we will be discussing the critical role of preclinical science in healthcare innovation. I will begin by providing some background on IMMR and to introduce the topic of preclinical science. We will then discuss the elements of preclinical research and the key outcomes of each stage, and then address the importance of choosing the right models for conducting preclinical studies with. Finally, we'll discuss some considerations for selecting a partner for the conduct of preclinical studies, and then we'll open it up for questions and discussion. In the development of new medical innovations, the preclinical phase, the preclinical science phase, helps to inform and perfect the design of new products and demonstrate the suitability of products to advance to human clinical evaluation. Generally speaking, preclinical science includes preclinical studies, histopathology evaluation, and the training of clinical investigators on how to use new products safely and correctly in human clinical trials. IMMR is a preclinical science organization based in Paris with a 20-year history of working with medical device, biopharma, and biotechnology companies. Our services range from early stage through to GLP preclinical studies, histopathology evaluation performed in-house by our team of board-certified veterinary pathologists, and surgical and interventional training for clinical investigators. Our values, which we live every day, include making substantive contributions to medical innovation and human health, providing unparalleled technical excellence in preclinical research, assuring 100% customer satisfaction, remaining objective and impartial, and at all times maintaining the highest ethical standards and concern for animal welfare. We have sophisticated facilities, including 5 standard and hybrid operating rooms, and advanced capabilities like cardiopulmonary bypass, state-of-the-art imaging modalities, and a telecommunications and data streaming platform called IMM Remote that allows teams to participate remotely in studies conducted at IMMR in Paris. We're a busy lab and have accrued world-leading experience with both cardiovascular as well as non-cardiovascular new therapies. And thus far, new therapies that IMMR has helped advance have been used to treat more than 1 million patients. So that's who we are. Our panelists today will include Doctors Nicola Borenstein, Luc Baer, and Laurence Biet. Nicola and Luc are IMMR's co-founders and senior-most study directors, and Laurence heads our pathology services division. And just to talk for a minute about the importance of preclinical science in general, the earliest preclinical studies help to validate the basic proof of concept and procedural safety in using new technologies and begin to inform elements of product design. Ongoing studies help to perfect design of new products, associated delivery systems, and implant procedures, and begin to inform the biological effectiveness of new technologies. And finally, GLP studies help to document the chronic safety and performance of new medical technologies so that their approval to advance to human clinical investigation can be justified. Now, the results of preclinical studies are certainly of vital importance to management teams, but preclinical research is not just an inwardly facing activity. Indeed, there are important external stakeholders as well. For example, regulators and institutional ethics committees, that need to provide approval for new products to be able to be used in human subjects, thought leaders and clinical investigators whose support is needed and who need to be willing to use technologies in their patients, and of course, funding agencies and investors who need to decide whether to finance these activities, and if so, at what valuation. Now I'd like to introduce Luc Baer, who will talk to us about the various phases of preclinical research in more detail. Luc.
Well, thank you very much, Rob, for your introduction, and good morning or good afternoon to everybody. I am Luc Baer, and I'm one of the scientific director here at IMMR and one of the co-founders. And I will be in charge today of talking about all the preclinical pathway and the full realm of activities and steps that you have to go through to validate your device from the idea to the passage to the human use. These first slides give you an overview of all the activities that you have to undergo to make sure you can progress from one step to the other until the final goal, that is a human application. The ideation of the study and the prototype or device that you intend to develop is usually a mixture of idea between a physician, engineer, with a lot of brainstorming on the conception and the potential application of a new medical innovation for a disease application in humans. This is usually very early before entering any animal testing, and that is usually done within the team of our partners. They usually come very early to see us to then talk about this preclinical pathway that goes from proof of concept, feasibility and R&D studies, then regulatory studies, and before entering the human application and use, regulatory training of the end users. Here you see described in this picture that the different phases, they are all very important, but one of the critical steps would be to translate a very raw device that you use in the proof of concept into a very refined device that you would be using in your GLP study and then in the end in your first in man. In the middle of that, there is a feasibility study. And as you see described here, during the feasibility study, we develop all the different iterations of the prototypes, refining, improving. Each time that you have a development of a prototype, it has to be tested into an animal model. If it's a success, and if it's a reproducible success, then you can move forward. But if you have adverse events and problems that you encounter, then you have to go back to the engineering, redesign, and then come back to the animal testing and make sure that this new design can be effective, and so on until you have a reproducible and efficient and safe device that can enter a GLP study, and then the training phases. This whole realm of activities, I will now describe a little bit more in details, everything that has to be done at each step. The next slide here is about the proof of concept studies. As I was mentioning, this is directly after the ideation. When partners come to us, they often have a very raw prototype in their hands, and they ask us, will this work in an animal model? We always say that we have to be creative in these proof of concept studies because it can very rapidly end up into the death of a project. If you are not creative at this phase with a rapid prototyped design of a device, you would most probably have difficulties in implanting that in an animal and making sure that you can have very early information about the potential of the device itself. Here, the proof of concept is a lot about the potential and the early demonstration of it. It contains science, of course, because you will be implanting your device and making sure that this early prototype can work, can provide some efficacy, and at the same time, some very preliminary idea of some safety. And if you cannot obtain this early information, then you might be in a difficulty to obtain further financing and to move forward in the step of the development of your device. So proof of concept is critical, because if you fail in the proof of concept, you might just be at the dead end road of your device development. And for that, you have to get the help of people with expertise, trained and creative, as I was already mentioning, because you have to make it work in an animal with a very raw device at the very beginning. The next slide is about talking about the feasibility studies. I was saying that the proof of concept is very crucial because you can end up with a dead end. In the R&D phase, we really expect that you encounter the problems. And in the R&D phase, you have actually to address all the problems before going to the regulatory study. And in this phase, failing should not be a problem. Failing is what you should expect, because when developing complex devices, there has very rarely been in our hands devices that goes from proof of concept to regulatory study. This would be a dream, but it's not reality. In this feasibility study, you have to expect adverse events, you have to expect problems that you need to solve. And it's this phase that is there to help you, making sure that the iteration, that the implementation and the improvement that you bring to the different prototypes of your device are going to be reproducibly efficient and safe. I am here mentioning some numbers. I'm talking to about 4 or 6 cases, each iteration of prototype. Why do I mention this? It's because when you test a prototype, you cannot conclude about the safety and the efficacy potentially on 1 or 2 cases. Making so quick conclusion might just give a no-go to a prototype within this feasibility study. You have to imagine that during this feasibility study, there is also the development of the model itself. So the failure can be 100% the device, but it can also be a mixture of device failure, but at the same time, complexity of development of the animal model, and also complexity of developing the way of implantation, the method of implantation. That's why I'm saying here, each iteration of prototype should be tested in approximately 4 to 6 animals to make sure that we conclude to the right problems. Is it the device that needs to be improved? Is it the animal model that needs to be changed? Or is it the methods of implantation that needs to be improved? Then during this feasibility study, you can have multiply the iterations and the testing of your devices. And as I was mentioning, if you encounter a problem, you go back to redesigning and you come back to the animal model to retest it. In the end, if you obtain a device that is successfully implanted and reproducibly implanted in an animal model with good results, you can start thinking about going to the design freeze of your device. The design freeze really means freezing the R&D at these steps with an almost fully accomplished, refined model of your prototype, and at the same time, of course, being sure that the animal model is a good one, and the full setup of animal experimentation should be confirmed at this step. When I talk about the design freeze, I'm not only talking about the implant itself. You should also make sure that your delivery system is fully designed into its final version, because the delivery system has to be tested, of course, also in the regulatory study. Why am I so strong on making sure that you bring to us your final version when entering the regulatory study? It's because after that, when we initiate the regulatory study, you will not be able to change the design of your device or the design of your delivery system. All that has to be frozen because the regulatory bodies will not accept a study in which you change the iteration of your device in this regulatory step of the validation of your device. The next phase, as I was already starting to talk about it, is the regulatory phase of this preclinical evaluation in an animal model. It is crucial that you make sure that you enter this phase with 100% confidence that you have the final version. And this full regulatory study should be compliant with the regulatory that is in place in the continent or in the country that you are supposed to apply for approval. I'm talking about, of course, Europe here, because we're in France. We are talking about the USA. We are talking about China, for an example. These different agencies in these different countries or continents have their own requirements. Even if I have to mention that there have been efforts that the requirements are homogenized over the different countries and over the different continents. However, there are tips and tricks and there are particularities for the FDA approval, for the CE mark, or for the Nmpa in China. Talking more about that, all of them need good science in these regulatory studies. And good science is making sure that you design your study with enough animal per group and that you involve, if possible, a control group in all these regulatory studies. At the same time, it is more and more required that a diseased model is also implanted at the favor of these regulatory studies. A regulatory study design should include healthy animals, and as I was mentioning, having an effort to include at least a small group of diseased animals. And when I'm talking about a group, you have to make sure that the group is properly respected. A group is composed of the same animal model, of the same design of your test article, your test medical device, and of the same exact follow-up. You cannot mix a group with different follow-ups or different designs or different control test article. Each group is composed of at least 5 or 6 animals to make sure that you can conclude with enough relevance in terms of science and relevance of the results for the safety and the efficacy of your device. The follow-up of these regulatory studies have to be according to the different devices. The longer you can, the better. Usually the FDA, the CE mark or the Nmpa ask for different follow-ups so that you can address what it is so-called the acute, the subacute, the chronic efficacy and safety of your devices. And that has to be discussed according to the different prototypes or different applications that we'll be developing. The key outcomes is, of course, obtaining data on the chronic safety and performances. The complete data set and the full report for the regulatory submission should be described in the final report. And actually, what the regulatory agencies require from us and from you is a full file containing all the data of this phase of the regulatory study with the full evaluation included into this documentation. At the end of such a regulatory study, before entering into the clinical applications, you have to make sure that you properly train your physicians and the end users about how to implant these devices in the humans. And for that, please make sure, for the safety of the patients and also for the success of your device, to make proper training of all the teams that will be first using your device in the clinics. Why do I insist on that? Of course, I insist on that for the safety of the patients. You cannot imagine that a complex device is transferred to clinics without practicing. And for that, usually we train the physician with the full medical team being invited in the lab to perform several implantations in an animal model to make sure that they have in their hands a full rehearsal, real condition in the OR of what they will be doing in humans. And I will want to also insist on the fact that the proper training makes sure that it is going to be safe for the patient and that you're going to have a success for your first in man and for your clinical trials when developing and then marketing your devices. And this is crucial for the patients, but also for the success of your studies and your medical devices. Thank you very much for your attention. And I would like to pass on the speech again to Rob for the next speaker. Thank you.
Thank you very much, Luc, for taking us through that series of steps within the preclinical research pathway. And our next presenter will be Dr. Laurence Villette, who will describe for us elements of pathology evaluation. Laurence.
So thank you, Rob, and good morning, good afternoon, or good evening to all of you. So I'm head of pathology at IMMR, and I would like to try to open a little bit pathology black box. So pathological evaluation is final assessment of a preclinical study at the end of the follow-up, or for animals who are early sacrificed or who died prematurely. It is mandatory in GLP studies to address two critical questions. First, the local tissue tolerance, then the systemic tolerance, and potential adverse effects at distance from the site of implantation. But pathological evaluation can be also highly informative in feasibility studies, so in the R&D and feasibility preclinical life of a device, as Luc mentioned before. Pathological evaluation includes microscopy at explantation and histological assessment in tissue sections. Our role is to evaluate, to describe, and to illustrate. So first, macroscopic evaluation of the device and the site of implantation. The device and surrounding tissue must be systematically and carefully inspected to observe the position of the implant, but also the tissue reaction, so healing, ingrowth, fibrin deposits, mineral deposits, and any change that we can observe with naked eye. Here are a few examples of what we can see. So in A, bioprosthetic aortic valve. In D and E, you can see some fibrin deposits around the chordae of the mitral valve or along the device passage. In B, injuries to the mitral valve due to the device. And C and F show some animal models of myocardial infarction in the right heart in C and in the left ventricle in F. You know that calcification of the bioprosthetic cusp is a major issue in bioprosthetic valves. And this is more accurately assessed using high resolution radiography, so faxitron or micro-CT. We can also observe with faxitron fractures or deformation of the metallic frame of the device. So here are two examples to illustrate this point. On the upper panel, you have an example of very calcified bioprosthetic valve with those three roots at the commissure of the valves on the whole of the faxitron of the whole device. And in the right part of the slide, minimal to marked calcification of the removed cusps. On the lower panel, you can see obviously a fracture, at least one fracture of the stent. The second part of the macroscopic evaluation at explantation is the examination, weighing and photographs of the major organs after full necropsy. Standard samples and samples for macroscopy findings are taken and will be submitted to histological evaluation. A few examples again. Of course, we look for a sign of thromboembolism. So thrombi, emboli, or infarcts in the downstream organs. You can see two examples of renal infarcts in A and B. So in A, it's a recent one because it's still hemorrhagic. And you can even see the causal embolus at the limit between the cortex and the medulla of the kidney. In B, it's an older infarct because it's whitish, fibrotic with depressed surface. We also look for thromboembolism in the pulmonary arterial tree when we implant a device in the right side of the heart. And indeed, you can see a fibrinous embolus in these arteries. We see this from time to time. Now, some technical considerations. most cardiovascular implants are very bulky, complex structures with metallic frame, with bioprosthetic tissues, with fabrics. And in order to obtain section from the device and from the surrounding tissues, so to preserve the intimate interface between the device and the local tissue, we have to use highly specialized histological techniques such as resin embedding. And those techniques, you can imagine, are highly specialized, need highly specific equipment, know-how, and some adaptability. I won't go into much detail about technique, but the first step is usually to make slices from the device and surrounding tissue with benzo, for example. And this trimming step is really crucial to me for two reasons. First, because trimming makes possible to observe the device and its relationships with the surrounding tissue that is not accessible before trimming. We take many pictures from this step because secondly, this picture will guarantee the continuity between the 3D complex structure of the device and the 2D histological sections that will be evaluated by the pathologist under the microscope. To illustrate this point, here is an example of, again, a bioprosthetic aortic valve, same one. And in this study, we performed 3 coronal sections from the device and tissues at the inflow, commissural, and outflow level. And you can see the aortic wall, the small black pieces are the stent struts. You have probably noticed in the commissural section that the bioprosthetic cusp have been removed. You can see only the roots of this cusp. So it enables me to mention that, of course, routine paraffin histological techniques are amenable to make section from the soft tissues, so from the cusp, for example, or from all the samples we took from the organs. Now, what do we do with these sections? So the histopathological evaluation aims to assess different parameters. For local tolerance, of course, there is a local tissue reaction. Most implants will be progressively incorporate into a fibrous tissue. So this is tissue in growth, pannus or neointima. The pannus should be recovered by endothelial cells to prevent thrombosis. An inflammatory reaction is also expected and needed for tissue healing. But we do not want to have much fibrous pannus. For example, in endovascular stents, we do not want to have restenosis because of the pannus. We do not want to have much inflammation, fibrin deposits, or mineralization. That's the reason why the evaluation with histology is not only qualitative, but also semi-quantitative, using a 5-grade scoring system to estimate the severity of the changes. And when possible, some parameters can be also measured using isomorphometric analysis. So let's move to a few examples now. The first one is a simple one from an aortic stent. And on the high magnification on the left, you can see a resin section with, in black, again, the stent strip. Now you can recognize them. The fabric, the media of the aortic wall. And you can see this diffuse fibrous tissue that incorporates the device and will stabilize it. And the fibrous pannus is covered by endothelial cells on the luminal surface of the pannus. In this picture, I don't see any inflammation. In the next example, from a sheep now, the aortic stent was implanted in a pig. In this example, again, the 2 stent struts from the stent, the fibrous pannus, the fabric, the endothelium. But here we have an inflammation. We have first mononuclear inflammatory cells, so lymphocytes and macrophages in picture B. And we usually see also multinucleated giant cells, mostly around structural material or fabric, so right fibers. Now, a few examples from bioprosthetic cusps, so from paraffin sections. In these two examples, you can see the parallel between the macroscopic features of the cusp after removal, the faxitron, and the corresponding histological sections. A bioprosthetic cusp is made of pericardium, so it's a yellow layer of tissue you can recognize here. I should say that I've chosen two very bad examples. Between the first one is an example of the highly calcified valve, highly calcified cusp. You can see this calcium deposits either in the H&E and S staining, but also in the special stain for calcium, which is alizarin red. So in red, you have those calcium deposits that will, of course, lead to structural changes of the cusp. On the lower panel, an example of an infectious endocarditis. On the cusp, a lot of fibrin, calcium also, and the gram stain shows gram-positive bacterial colonies. Now, the report, the pathological report, should give a summary of findings by groups. We do not need description of individuals, but we are in an experiment with animals, with groups of animals. So we need to have the summary synthesis of findings for the groups. Our philosophy at IMMR is to deliver a lot of annotated microphotographs, of course, to illustrate the report, but also to use this photograph to make the reports more understandable for a non-specialist, to present the data and discuss the data with the sponsor and study director. And we love to share our expertise with other scientists. The report should conclude about the safety of the medical device in this animal model. So about local and general tolerance. But we have to answer another question. Are the findings related to the device or not? Are the findings now, finally, are the findings expected findings in the context of this study, in this animal species at this time point? And all this interpretation is based on our experience as veterinary pathologists of medical devices, but also in comparison between control and test groups if present. I mean, test and control groups if present. Okay, so of course, pathologists are a very important person, but a pathologist to me is a full member of a team of experts, because pathological evaluation, pathological data should be correlated to clinical, to biological, to imaging data. And I would like to conclude that the interpretation of the findings of all the results are depending upon the animal model, the species, all the conditions of the study, and specifically on the species we have chosen, with the advantages of each species and limits of each model. Thank you for your attention.
Thank you very much, Laurence, for describing the elements of pathology evaluation for us. And our final presenter will be Nicola Borenstein, who will talk to us about considerations regarding selection of animal models for use in preclinical studies and some considerations for selecting a preclinical laboratory. Nicola.
Well, thank you, Rob. So what I will do now is speak a little bit about animal models, different animal models. And let's just put it this way. A question that we get very often is, should I perform my study on a sheep or on a pig? So what's it going to be, a pig or a sheep? Now, of course, the issue is a very wide issue, and we could write a whole book about it, but I will try to tackle a little bit about those concepts. What is the best species for your study? So the first thing I want to say, and the first message I want to convey, is that size is not everything. There are scientific considerations, technical, practical considerations as well. And just to illustrate, we will look basically at the pros and cons and the advantages and limitations of two typical large animal models that we use here at IMMR and that are commonly used for large animal validation. I will give you a couple of examples. This is not the gospel. I am not trying to convince you that one is better than the other. I'm just trying to convey a couple of key messages. So let's look at pigs, for instance, and let's look more particularly at cardiovascular research. Now, what I will say is that the heart anatomy is closer to that of humans. And we know a little bit about that because we have led in the lab internal studies that are being published right now on comparative anatomy of the heart, and in particular of the mitral valve and the tricuspid valve. And this was done by two of our younger colleagues who have joined a couple of years ago and who have increased deeply our knowledge. And so that is Alexis Morlet and Olivier Chevènement. So what I will say about, say, the mitral valve, well, in a pig, it is thicker than in a sheep. And the valves, the cords, the coronary system, everything looks a little bit more like a human. There is a right heart dominance, which is much closer to what you would find in a human. Other aspects are interesting as well. Transesophageal echocardiography is better in a pig than it is in a sheep. Those animals are easily sourced at any weight, and they are monogastric, meaning they have just one stomach like we do, which means that you can give them drugs per os, which is a lot more difficult in a sheep, which has something like basically 4 stomachs. It's a little more complex than that, but for all common and practical purposes, they have 4 stomachs. And whenever you give them a drug, it is basically destroyed per os. Now, other aspects of sheep, which make them particularly interesting for the studies we lead here, there is more space around the mitral annulus. So for transcatheter approaches to the mitral valve, it is a great advantage. Those animals do not grow when they are at, say, 60 to 80 kilos. Basically, the heart will not grow anymore. They're adults, and they can be used without having the fear of them outgrowing the device. So they are typically used for long-term studies. One very important aspect is that they're very robust and docile animals, but more particularly robust. So basically you perform an interventional procedure or maybe even a cardiac surgery on the cardiopulmonary bypass in the morning, and in the afternoon they're standing and eating. So that's a great advantage. And another aspect that is very often used with benefit is their tendency to calcify, especially when they're young. And this intense calcification is a way of challenging the valve, and that is seen as an advantage for regulatory studies. If we look now at limitations, well, it's pretty much the same thing, but the other way. Now, pigs grow very rapidly, so they might outgrow the device. Those animals are also more susceptible to infection. They are more fragile in general. They have a higher incidence of arrhythmia during anesthesia. They do not die systematically of arrhythmia like I have heard in the past. That is not true. And in a laboratory that has good experience with anesthesia and critical care, basically animals should not die on the table. But it is true that the arrhythmia is more marked in pigs than in sheep. They are also more difficult to handle. They're very strong animals. They're basically all muscle. So whenever you do a surgery, you have a lot of muscle to cross. Likewise, their heart is very hypertrophic. So that can cause some problems whenever you need space in the ventricle. and their inflammation is certainly more marked than in ruminants. And finally, in sheep, the one thing that I might want to stress here is that that junction between the aorta and the mitral, so the so-called aortic mitral curtain, is practically non-existent in a sheep. And that can cause a lot of problems whenever one implants a heart valve, especially transcatheter heart valves, in the aorta, so for TAVI, or in the mitral position, in an orthotopic position. So there can be conflicts, anatomic conflicts. Those animals are also more difficult to source, and they are more expensive. So what should we keep? What's the take-home message here? Basically, you can use both sheep and pigs for any of your large animal studies, but there are some particular aspects of each species that one has to consider. Now, let's get out of the realm of strictly cardiovascular research, and let's go maybe in more dermatology or urinary, or maybe laparoscopy. Then definitely a pig will be preferred. If you absolutely need chronic studies, say over 6 months, then definitely you will tend to go towards a sheep, but both are possible. Now, there are other species that are used, of course, such as canines and bovines. In a nutshell, bovines are used for total artificial heart and LVADs, and dogs can be used for a lot of things. They're not used as much these days because of ethical concerns. Those animals are pets. But they have anatomic particularities that make them very much used for left atrial appendage ablation, EP studies, sometimes renal, urinary, and neuro. And you can see here an image that I just want to point out that already Laurence has shown before, but it shows you how strong the calcification can be observed in a sheep. This is a sheep. So on the left side, what you see is the valve with no calcification. On the right-hand side, you see it with intense mineralization, and that is just within 3 months. Other aspects are also very important, not just for everything that I've mentioned, but also for comparative anatomy and comparative imaging. As you can see on the left-hand side, you have a sheep, a swine in the middle, and a human on the right-hand side. And you can see that those images are pretty much mirror images. So the fact that the heart does not have the same orientation as that in a human will imply a certain number of things in terms of imaging. And so you want to choose a lab that has good knowledge of this. Likewise, you can see that the height of the atrium on the left-hand side, those images are CT scan, of course, especially in the sheep, the height of the left atrium is pretty much half of that of the ventricle, while in a human, it's almost the same. What does that mean? It means that whenever you bring a device in the left atrium in a sheep, there will be a problem of real estate. You will not have the space that you need. That has to be known in advance. I'm not saying that those limitations make it impossible to use sheep. I'm just saying that it has to be known and it has to be addressed properly. Here again, an interesting image that shows you the distance between the esophagus and the left atrium. In a sheep, it is tremendous. It's basically 5, 6, 7 centimeters, while it's just a couple of centimeters in a human. So real in situ transesophageal probe can provide excellent images in a human. Well, it's very difficult in a sheep and a little less in a swine. So we have approaches for that, which kind of counteract the fact that those images are so difficult to obtain by putting the probe directly against the left atrium. So again, as you see, you have beautiful images here on the left-hand side. You can see the left atrium, the mitral valve, just at least as nicely as you would in a human. The only thing that I will say is that you cannot put the probe exactly on the same spot. Those are basically tips and tricks that we have learned, sometimes the hard way, over the years. There are some other images also that can be provided in an animal lab, and I think that people should be made aware of that, and they're extremely insightful. You can see here that the images are provided directly with cameras that are introduced inside the left atrium on the left-hand side or in the left ventricle on the right-hand side. Those images are real images, of course, with high-speed cameras. This is why it's so slow, of a heart that is beating spontaneously. So we have to replace the blood by saline. It's a very complex endeavor, but what we get is spectacular. And you can see every single bit of movement of the mitral valve. You can get that through imaging, as you see in the middle, with even quite refined approaches, such as this one through a Philips machine, but it just cannot beat the camera stuck inside. Again, some images that I wanted to share with you this morning to compare the differences between human, sheep, and swine. And that has to do with the aortomitral continuity, which you can see here in a sheep. The anterior leaflet of the mitral valve is pretty much on to the aortic annulus. Well, in a human, you have something like 1.5 up to 2 centimeters. That makes huge difference for TAVI. As you can see here, you are looking from the ventricular side at a TAVI valve that was put in situ in the orthotopic aortic position. And you can see that it is barely too low. It is pretty much on the right spot. But as soon as you raise the anterior leaflet of the mitral valve, well, you can see that there will be paravalvular leakage. And that is due to those anatomic constraints. Other things that one has to keep in mind is the frailty of tissues. You can see on the left-hand side, once we had done a mitral implantation, we removed the valve, and there is an almost complete destruction of the native anatomy. On the right-hand side, what you can see is that a TAVI valve has torn almost completely the anterior leaflet of the mitral valve. So again, I am not saying that those animal models should not be used. Quite the contrary. I think they should be used, and they are, in a way, sort of a worst case scenario. But you also have to know there are limitations. It is not because you get such horrible results in an animal that it will be the case in a human. Having said that, if you have so much destruction in the tissue, you probably want to address your anchoring strategy. A couple of other aspects that are also interesting, comparing animals to humans. You will see that the hemodynamics are not quite the same. The take-home message here is that the contractility inside a swine heart is very high, much higher than a human, and even much higher than a human with a left heart pathology. So a healthy sheep, and even more so a healthy swine, will be very contractile. What does that do? Well, it can break, it can cause fractures of stents, and that also has to be known. Now, I would like to move on to another aspect that Luc has mentioned a little earlier, and that is the fact of using or not using pathologic models. It sort of stands to reason that a diseased animal model should be the best platform to assess safety and efficacy of a novel device. But the one thing that I want to tell you is that coming with a reliable pathologic model in a large animal is extremely challenging. Not that it is difficult to create a lesion. Creating a lesion is never an issue. You can always tear a leaflet or sever a cord or cause an infarct. But the challenge is getting enough of a problem, but not too much of a problem. So enough dysfunction, but not too much dysfunction that you lose the animal, which becomes cost prohibitive because you have to develop a huge number of animals and animal models in order to provide a decent number. But more importantly, it is simply unacceptable from an ethical standpoint. You cannot develop a model on 100 animals just to get 10. So the challenge is just to get the right amount and to know what is sufficient, but what is not too much. And so you want basically a survival rate that is acceptable. So I will present just really quickly one model that we have developed in the lab, and which is that of the ischemic mitral insufficiency, which is of particular interest these days. So we come up with a very robust model that has pretty much an 80% success rate, meaning that 80% of our animals survive. And how do we do that? We inject inside the coronaries microbeads. And we do that once, twice, or 3 times, sometimes more, but rarely more. And what we want is to get enough dilatation, enough effect, which would be to have poor coaptation of the mitral valve, of the mitral leaflets, having no clinical sign of heart failure. So those animals are treated for their heart failure with ACE inhibitors and diuretics. That is what we consider an acceptable model. You can see here 2 CT scan views that show you the dilated and non-dilated areas. And you can see here healthy ventricle on the left-hand side and the diseased ventricle on the right-hand side. And what you can see is that the ventricle is very dilated and that there is major regurgitation. This is exactly the sort of model that we like to develop because those animals had no overt clinical signs. And we are doing currently exactly the same thing for the tricuspid valve. Those 2 models are being published as we are speaking. Here is the final image that I want to share because I think it is extremely illustrative of the sort of MR that you would get thanks to this serial embolization procedure. And now I will move to a final topic, which is also related to the concept of animal model, but which is that of how would you choose the best preclinical science partner possible. Now, I'm not here just to tell you that we are the best in the world, although I do think so in a way. But what I want to convey is make sure wherever you go, whether it be in Asia, in North America, in Europe, or wherever else, that the lab that you work with has the highest ethical standards. You want truthful commitment to animal welfare. I think this is the #1 aspect that I think we should all consider. The second aspect I want to stress here is complete respect of confidentiality. If you go to a, say, a more university setting, and I'm not saying that you will not get the service that you need, but confidentiality and scientific independence, free of bias, might not be guaranteed just as well. You want a lab that has thorough knowledge of comparative anatomy and physiology to select the best model, yet understanding their limitations. And this is pretty much what I have shown beforehand. I think it is important to be surrounded by individuals and a lab as a whole that has a problem-solving ability, because the long and strenuous path to commercialization is fraught with difficulties, and you will have lots of difficulties that can be solved, but you need to work with a lab that wants to do it and that wants to be on your side. That lab also needs to be a state-of-the-art technical platform such that the facility is familiar to surgeons and interventionalists, so that they feel in an environment that is very much akin to what they will use once they go to the clinics in their first-in-man study. So those are basically the messages that I wanted to convey. We'll be happy to answer any questions at the end, but I will thank you for your attention and pass on the floor to Minneapolis now. So thank you very much for your attention. Thank you, Rob.
Nicola. So to summarize, the preclinical phase or preclinical science provides essential information about product design and performance at the scale of human anatomy and is critical for ensuring that clinical trials of these products can be performed safely, correctly, and with compelling outcomes. It's critical that studies are designed with appropriate scientific rigor and conducted in appropriate models at facilities with requisite capabilities and experience. And histopathology evaluation, while certainly a requirement in regulatory studies, as Dr. Fiat explained, can provide valuable insights all along the development pathway. So the preclinical phase is indeed more than just checking a box. It is a crucial and significantly value-enhancing phase of product development. If you're interested in learning more, our team just recently contributed a chapter on preclinical science to the newly published edition of the PCR textbook, Percutaneous Interventional Cardiovascular Medicine, that you can read. Or better yet, you can get in touch with us through either email or LinkedIn or our website to connect with us and discuss your preclinical needs directly. And finally, while the pandemic is ongoing, wanted to let you know that IMMR remains open for business. Sponsors are welcome to come and join us in our labs in Paris and can do so safely, or teams can work with us directly using our IMM remote platform. So thank you very much for joining us today. Thank you everybody for watching. We do have a few more minutes available for some questions, and we'll get to those right away. We have some regulatory questions, we have some technical questions, we have some business questions. Our first question will be for Luc Baer. And the question is, hello, thank you for today. It's great to hear that you're qualified to conduct studies according to US GLP standards. But is this standard accepted by all regulatory agencies worldwide?
So thank you, Rob, for the question and actually the audience for the question. So the regulatory environment in which you perform your preclinical studies are a bit different in the US, in Europe, or for China. There are the GLP from the FDA, the 21 CFR Part 58, that really give the requirements in which the study have to be conducted. And there is also the GLP from the OECD, which are a bit different. However, if you perform your studies according to the GLP from the FDA for the regulatory part of it, and according to the GLP from the OECD, according to the regulatory environment, yes, it would be accepted. What is to be known is that this is only the environment in which you perform your study, the regulatory environment. Inside the study, to be accepted, you have to make sure that the design is properly performed. And for that, being the US, being Europe, or being China, science that is inside the regulatory environment has to be optimal. And for that, you have guidance, can be the ISO. You have also the expertise of the center of research with which you perform your study. And that is of a tremendous importance. Regulatory-wise, it's only the envelope, but what's inside has to be addressing all the potential questions that can be raised on your delivery system and your implant. So that is very important, and that lies within the hands of experts because it's always different from one device to the other, and has to be discussed, actually.
Thank you, Luc. So Laurence, in a related question for you, do FDA's GLP pathology requirements differ from those of other regulatory agencies, for example, the European notified bodies or the Nmpa in China? Do any countries have their own unique requirements?
Okay, so thank you for the question. The answer is no, there is no, I mean, the requirements are pretty similar for the pathological part of the studies. We have to provide very precise, very well pathological data from a board certified pathologist. And we have to have all this data in a proper manner, I would say in a scientific manner, to show that the local tolerance and the general tolerance are good, and also to show that we have done the study, the pathological study, the right way with all the methods we can use to have this information. So no, there is no very specific requirements.
Perfect. Okay, thank you very much. Nicola, we have some technical and scientific questions that I will pose to you. And they are the following. There are numerous anatomical, hemodynamic, and metabolic differences between animal species, as well as between animal species and humans. My question is, how reliable are animal models, especially for newer techniques such as TAVI or valve-in-valve procedures and so forth, regarding not only the operative procedure in se, but also concerning the histologic modifications, propensity to infections, inflammations, etc. Next, how important do you consider the study of comparative anatomy? And I know you've touched on various aspects of these questions. And a related question, which species would you prefer for cardiac, coronary, and microvessel studies with a non-remaining device, with a microcatheter and or a guide wire? Nicola.
Okay, thank you, John. Thank you. It's kind of, something I have mentioned before. There are many different models that can be used. There are pros and cons to each. I've only mentioned here basically sheep and pigs. I have very quickly mentioned other species, but it is true that comparative anatomy is key. And whenever I give this talk, or whenever we have a new group that comes and asks exactly these sorts of questions, my first answer is that the three most important aspects will be anatomy, anatomy, and anatomy. So it's not just me. In the lab, we're very keen on that. This is something we like very much. And comparative anatomy is key to the understanding of the limitations of the model. So, I tackled a little bit about this where I showed fractures, where I showed tears in the anterior leaflet, etc. This is a typical problem of animals. So it's a limitation indeed. And if you don't know that, if you think that the problem you have in an animal will be a copy and paste in a human, you're probably wrong. And so knowing, for instance, you mentioned TAVI, the angle between the aorta and the mitral, that's another aspect. There's a size aspect, and then there is also a dynamic aspect, and there is a comparative anatomy aspect. So basically, in a sheep model, the aortic annulus and the mitral annulus have this sort of angle. In a human, it would be like this. So whenever you implant a TMVR, you end up with a lot more potential LVOT obstruction in a human than you would in an animal. So that's it is a limitation of sorts. It's actually, as opposed to what I said before, it's not a worst case scenario. It's just that animals are a more favorable environment. And so how do we know this? We know this because we study comparative anatomy. So yes, the answer is absolutely. Some animals, I think this question was asked as I had not yet presented the comparative anatomy aspect. So I think what I said earlier addresses the question. Of course, we could speak for hours about the interest of dogs or goats, and there are other large animal models that can be used. But then there are other aspects that I have not necessarily mentioned a lot, like price, like sourcing, like ethics, like the price to house such animals. I mean, there are many other aspects than just, anatomy. Having said that, I think it is key. And to link it to the second question, which is which species would we use for cardiac coronary? Well, in the article, in the chapter we just published, we have a a large chapter about this, where we mention what models have been used for the coronary. Truly, I think most people these days, when they think large animals and coronary, think pigs, but we have also implanted coronary stents in sheep, in dogs, and they all have limitations. The first thing that we usually look at is size. You want to have a fairly large coronary vasculature to mimic what you would have in a human. The truth also is that in pigs, you probably have a more contractile environment and a more, I wouldn't say inflammatory, but you will have more fibrin, more probably neointima. I mean, you will have a different kind of tissue reaction. Now, of course, Laurence would answer this better than me. But yes, there are differences. And I see that the question also has to do with microcatheter, guide wire. The truth is we have used all those animals, and we have used also dogs for that, namely for coronary sinus studies, where we have tried to advance a catheter that was going into the coronary sinus and retrograde all the way to shunts that are very far in the coronary vasculature. And dogs present advantages. They have a lot of collaterals, but the thing is they have too many collaterals. That's quite far from humans. So you can use one or the other. I'm not saying that one is necessarily better, but you just have to work with people that know what they're talking about in terms of that specific animal model and know where to go. Sometimes we start with a pig for one aspect, maybe for an acute aspect, knowing that this animal will grow tremendously and might outgrow the device. So we'll do the acute aspect, the acute delivery on the pig, and maybe we'll do the chronic on a sheep. Every study is different. That's what I can say.
Okay, thank you. I think we have time maybe for one more question. And Nicola, I think I'll address this one to you. It's a business question. There was also a question about whether the slides were going to be made available, and the answer is absolutely yes. And we'll provide some more information about this. So if your question was not addressed, we'll try to follow up with you individually. But again, feel free to contact us to talk about your needs specifically. And the final question, Nicola, is typically investors place tremendous value on human clinical experience. Is this always the holy grail? Can compelling preclinical data influence an investment or acquisition decision in a startup developing cutting edge technology?
Oh yeah, I think it's paramount. I have seen many different stories. We have seen people that have done very few animal cases and have managed somehow to go to the clinics, maybe because the regulatory environment at that point was fairly loose. And they managed to have their first in man and went on and actually were bought out. But in this day and age, I think that everything has to be done very properly. And I think you need to at least convince the regulatory agency. Well, you have to convince people from a scientific and medical standpoint that it is worthwhile. And then you have to convince people also on a regulatory level that everything was done properly. And you have to go through animal labs to do that. And I think that there is compelling preclinical data that will influence acquisitions. And we have seen many different stories. Many of the labs many of the startups that have worked with us have known beautiful exits. Some have simply disappeared for many different reasons. But whenever we manage to get those convincing, compelling images to show that it can work and that it doesn't hurt the body or in a relatively acceptable manner, then yes, it is convincing. And you're mentioning investors or acquisitions. It happens quite often that investors contact us. Of course, we never disclose anything if it's always something that is done in a very open way, where the company might say, well, I have this investor who wants to invest a lot of money or who wants to buy us out. They want to know what you think. And I tell them, oh, okay, I can do that if you'd like me to, but I will have to be very honest about it. I will not lie. I will not say, dismiss the failures we may have had. And usually that's the way they want it. And so, yes, the standpoint that preclinical science may have, I believe, is of the utmost importance.
Great, thank you very much. So Josh, I think I will turn it back to you. I want to thank our panelists for an engaging discussion, and certainly thank all of our participants for tuning in. Thanks very much.